I'm Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, I sat down with professors Danny Chou and Helena Safavi to learn more about cone-snail venom insulins. These novel proteins offer the potential to generate new medicines for diabetes and also inform on compensatory binding modes of truncated insulins. In addition, these proteins act as new tools to study insulin receptor signaling. Well, thank you both for agreeing to come on Exploration Science.
Yes, our pleasure.
Yeah, happy to do it.
Maybe first off, you could talk to me a little bit about how you both connected with each other.
I feel like Danny and I should have met before this meeting to go over our history together. I think Danny reached out, so we published the paper on the very first insulin from the cone-snail geographers. And then Danny had just been recruited to the University of Utah, or recently recruited to the University of Utah, and he can elaborate on that. And then he reached out to us and that we should meet and discuss this finding.
Yeah, so that was during my very first year starting my independent career. So I will say that's probably 6 months after I started, and then they have the paper published. And we had a internal discussion in our group meeting that we thought this is a very cool study, and then it's pretty worthwhile for us to further pursue. So that's why, at that time, Helena was still a postdoc in Toto Oliveira's lab. So I contacted Toto, and then Toto is a pretty special person in the way that, he didn't really use email by himself. So that took some time for us to finally meet together to discuss. But that was a great beginning.
Yeah, and Helena, you're now you're visiting the University of Utah right now. Can you tell me just a little bit about that, like what you're doing?
So I'm an associate professor at the University of Copenhagen in Denmark. So I was actually an assistant professor at the same department that Danny was at, the biochemistry department at the University of Utah. Then I ran into some immigration issues and left the country to go to Denmark. And I'm associate professor there now and looking into returning in the summer. Yeah, but I've always kind of kept an affiliation with the University of Utah and work closely with people here.
That's great. And then Danny, you've moved on now to Stanford. Can you tell me just a little bit about that transition?
Yeah, my official start date was literally 2 years ago. So basically that's April 2020, and that was a pretty difficult time for anyone to move professionally, also personally as well. But it was too much to change the start date. So I moved somewhere around summer, and then my lab moved around the fall. But it's been good so far. That was a little bit difficult in the beginning in the way that there's some lab renovation progress being delayed because of the pandemic for very obvious reasons. But it's been good so far.
Yeah. And can you tell me, Danny, still just a little bit, like maybe broadly, about your research group, the topics that you're studying right now?
Yeah. So in a kind of generic term, basically we are a group of chemical biologists interested in peptides and proteins. So things that more in the macromolecular level. That's why we get to know you early on when we started to purchase peptide synthesizer in my very young group at that time at Utah. Actually, the funny thing is, I think probably I started contact Toto, at a time pretty close to the time we contact you to set up the peptide synthesizer in our group, because that was all in the beginning of my independent career. So we are basically organic chemists in heart, in the way that we make molecules and then try to make molecules with some focus for therapeutic development, or make molecules such that we can study some of the interesting biology. So I would say half of my group right now is more on the therapeutic side, where basically we are trying to make a potential peptide or protein therapeutics better by using quite a bit of chemical modulations, and this example of venom insulin will be part of that. Another part of what we are doing is trying to make chemical probes, but in this case will be more peptide or protein derivatives, such that we can use that to study cell biology.
Nice. Yeah, you've come a long way. It's really exciting for me to watch your career, especially starting out so early together. So Helena, same question to you. Can you sort of just give a broad overview of your research interests?
Yeah, so our group is mostly a discovery lab. So we work on venomous animals, mostly venomous cone snails, although we've started to look into other venoms as well. And there are around 800 known species, probably a couple of, or maybe 200 unknown species of cone snails, and they all make different venoms, mostly comprised of small peptides. And so there's this really vast diversity of chemical space to explore. So we are trying to discover things that then we can carry on into either ideally in the future the clinic, or into molecular tools and pharmaceutical tools. So we are a discovery lab, which means we have evolutionary biologists, geneticists, one computational biologist. And then once we find something that we're interested in, such as the insulin or other interesting peptides, then we have a couple of pharmacologists who then study the activity of those peptides. And then we typically collaborate with clinicians or people with more physiology background to see if any of the peptides we have discovered is potentially interesting for clinical or preclinical development.
Yeah. So maybe you can tell me more about the work you and Danny are doing together on the insulins from cone snail. Can you just describe the use of insulins in cone snail venom, or their natural use?
Yeah, so I can answer that question, I guess. So the cone snail that we first discovered the insulin in is this fish hunting cone snail. It's a very unusual cone snail because it doesn't directly inject its venom into fish prey, but it opens like a fake mouth and releases venom components into the water, thereby sedating fish. So when we looked at the venom of that cone snail, we found insulin at fairly high expression, actually several insulin molecules. And that made us then look into the physiological function of insulin release into the water. And it turns out that just the presence of insulin in the water can make the fish hypoglycemic. So the fish basically experiences an insulin shock. So very low blood glucose levels, and that makes it easier for the cone snail to then go in and catch the fish.
So the insulin in the water, I mean, to get into the fish, is it so fast acting because of the way that they filter water for breathing? Like, is that the delivery aspect of it?
Yeah, so that does not really have to do anything with the fast-acting nature of the cone snail insulin, because you could also take human insulin, put it in water, fish swims in it, and then it starts, floating on the surface, and then you can add sugar, and the fish will recover. So there's most likely a mechanism by which the insulin enters through the gills, through the epithelial cells of the gills.
Yeah.
But it's not very well studied how it gets in, but it is well known that it does get in. And because it is so fast, it most likely gets through the gills into the bloodstream directly, rather than through the mouth.
Sure. And you did mention that the fast-acting nature is not a result of the absorption of the insulin. This is the specific nature of these insulins that the cone snails are producing. So can you, we'll talk about that, and then I'm going to totally backtrack and go just all about insulin. So.
Yeah. So maybe Danny, do you want to take over that part? So why the insulin is fast acting?
Yeah. So this ties back to the very beginning why we are interested in this whole thing is because this venom insulin has a very special molecular feature in the way that basically all the insulins, I would say all the way from human down to those other vertebrates in fish or in other species, they all have relatively long B chain. So I would say somewhere around 20, 30 amino acids. The last 8 is quite important because that's how insulin use it potentially to form a dimer first, and then 3 sets of dimer can form a hexamer. And this is a pretty common way for insulin to be stored, because in this case, insulin by itself is most stable. There are some cases where if there are some mutations there in the way that to avoid these kind of hexamer to happen, these normally will lead to insulin aggregation, and then that may ends up leading to beta cell death, and then that could be some special form of diabetes. So this is quite important. But in the case of the Venom insulin, there's no such fragment there. So it's a pretty short B chain compared to all the kind of evolution of native insulins from different species, and this is the reason why we think that this insulin should be monomeric, because it just doesn't have the fragment to form dimer and hexamer. And there were some studies in the past, pretty comprehensive studies in the 90s, to show that basically the rate of absorption for insulin after being subcutaneously injected is inversely correlated to the association state. So that means if it's more monomeric, you will get into the blood vessel faster. If it is, say, hexameric, you will get into the blood vessel slower. So this is the reason why at that time, even in the very beginning, just looking at the sequence of the Venom insulin, we had a feeling that this molecule should be monomeric. And then from the previous studies, if it's monomeric, we should get into the blood vessel faster. And then that's why we thought there could be some fast-acting properties here.
Yeah. And so you actually helped me, because I was going to have you backtrack and explain how insulin works. And you did that already, so thank you. So I'm curious to know, how small can you go so far with these, now making synthetic? So you're transitioning the natural product to a synthetic product. So talk to me about that.
Yeah, so native insulin has 51 amino acids, so 21 on A chain, 30 on B chain. In one of the versions we published in 2020, we call it mini insulin, mini because it has the shortest skeleton or minimized skeleton that needed to have the full activity. It's 43 amino acids. We also have a study that's not published yet, but I mean, this is relatively trivial for people who kind of understand insulin chemistry to know this. So the first 4 amino acids of the insulin B chain, it's not really required for any receptor engagement or binding. So we can delete that as well. So if we want to push it, we can go down to 39 amino acids that still have full activity, and then basically activate the signaling very similar to native insulin. So that's kind of the most we can push right now. But I would say that this is kind of mostly on just the insulin or insulin-like backbone. There are some pretty cool research directions in the way that people may use de novo protein design to create something in a way that you don't need those 3 disulfide bonds in between to have the protein. One of the very recent examples, not in insulin, but in this cytokine called interleukin 2, people in David Baker's lab at University of Washington, they can basically rewire the whole backbone, and in a way that still they will have the right residues, directed toward the right position in a way to activate the receptor. So I will say that it would be pretty cool if someone can do this as well, and then that will be potentially have something even easier to synthesize as well, because still chemically synthesized insulin is still pretty difficult to do, and then that's why most of the DNR do recombinant expression. But even with that, there's still quite some R behind it. So if one can have something that's pretty trivial to make, potentially this could help for the production of insulin as well.
And Helena, the insulins that these cone-snails are producing, It looked like from one of the references, the way they bind is different than the way that an insulin would bind to the human receptor. Can you just describe that a little bit?
So to go back one step further is like once we discovered the first venom insulin in this fish hunter, we actually cloned the insulin gene from many cone-snail species to look at what other species make insulin and what would their insulins look like. And fish hunters are there, of the 900 or 800 known species, only about 150 hunt fish, and the others mostly prey on worms. And some prey on other snails, including other cone snails. So many of these species also have insulin, and several other fish hunters also have insulin. And when you look at the insulins, they look quite different. So it's only the fish hunters that make an insulin that looks like vertebrate, like fish or human insulin. And within those, none of them have that B chain extension that Danny was talking about. So we assume that most of them would be monomeric or really not form a strong dimer, if at all. And then we started to look in the functional interaction of some of these insulins with the insulin receptor, and also recently the structural interactions, and saw that they basically, if you lose that B chain extension, you lose activity at the receptor. So human insulin no longer binds. And then the cone-snail insulins or cone-snails have evolved other residues, other amino acids that would then engage with the receptor instead. And depending on the species of fish hunter you find a different molecular mechanism of receptor activation.
Is there, with the fish insulin receptor versus the human receptor, is there also some homology there, or is it totally different binding modes?
Yeah, so at the structural level, the fish receptor has not been studied in detail, but when you do a sequence alignment, it is very similar to the human receptor. Different species of fish have different isoforms of the fish receptor, but it's typically very similar. So when we first studied the insulin, we had only studied it in fish, but because the fish insulin receptor is so similar to the human, we were quite sure when Danny, Toto, and I got together that it would also be active at the human insulin receptor. And then Danny tested it and found that it was surprisingly active, considering that it doesn't have that B chain extension that was believed to be critical for human insulin receptor activation.
Yeah, and Danny, that structural information, so I'm not a structural biologist by any means, I'm a lowly peptide chemist, but when you look at the structure and you see how molecules can sort of change orientation and make sort of surprising interactions, how does that help inform you of what you can accomplish synthetically? Or what, I guess, when you see something so different, how does that change your mindset about your methods?
Yeah, this is really a terrific question. And then this is really something that I would say beyond our surprise as well, how these molecules are teaching us, biochemically. So let's go back to the beginning. When we first started the project, we thought this is a very interesting molecule. And then we thought, because of the features I just mentioned, it's very worthwhile trying to investigate it. In the very beginning, when I was trying to pitch it to the postdoc to work on it, we actually view it as a pretty interesting natural product, chemical synthesis project, in a way that we'll see what's going on here. But like you mentioned, the cool thing, what we learned from, I'll say probably the two kind of more key peptide engineering papers we published, one recently, one in 2020, is that we can basically find some mutations from the Venom insulin sequence such that they basically increase the interaction with the receptor in a way that basically all the endogenous, well, I should say most of the endogenous insulins from all different species are not using. So basically it's through some sort of evolution way, in a way that snail basically discover these mutations such that we can change the way it binds to the receptor. And then really the key thing for us is that from some of the structures we saw recently, these new interactions may in the end change how insulin bind to a receptor and then kind of tweak the conformation of the receptor, such that very likely there will be some implication of how the signaling got activated. And then I will say this is something we are very interested in working on in the near future, because basically we know a lot about.
Sorry, could you say that again?
This is some of those things that happen.
We'll leave that in.
Yeah.
Yes.
So we know a lot about the insulin signaling pathway. If you go to some of those antibody companies, so like the one called cell signaling, you can find a very complicated map of all the protein components of the signaling. So it's probably one of the most well-studied one, if not the most well-studied one here. But really, there's very few we know about, in the very beginning of that charge, when insulin binds to the receptor, how does those arrows happen? How does the activated insulin receptor decides, which pathway they want to go, how they basically decided to go do more metabolic signaling instead of mitogenic signaling. I think this is still something that's very hard to understand. So normally I would like to use this kind of analogy. So in GPCR, because there are so many tools available, there are so many great structural biology efforts there. People have a very good idea of when you can bind to this part of the GPCR, you can have this sort of conformation. You may be able to bypass some of the signaling, so you can go for more biased signaling. In the case of insulin signaling, is still very hard. Or let's just go more broad. In the case of receptor tyrosine kinase, it's still not that further along. And but I think with the revolution of cryo-EM, for example, it's getting closer and closer for us to use insulin as a ligand, and then potentially peptide chemists like us to make all different kinds of insulin, trying to tweak the conformation of the receptor activated form, and in a way I try to understand more about the signal.
Right. Yeah, I love that. That was, again, you keep answering questions before I ask them, which I love, because one of my questions is what are the ramifications, right, beyond insulin when you're studying things like natural products, and so thank you for, again, jumping ahead and answering that. Helena, another question that I have for you is really about the cone-snails themselves. Are these, a couple questions, are they using this beyond just hunting? So do they use it for defense and like warfare against each other or other snails?
We don't think so, because of the large sequence library we generated, we could actually see that it's very closely correlates with predation or prey type. So you really see that the fish hunting cone snails make fish-like insulins. When you align the sequences, they're surprisingly similar to fish insulins. And then the snail hunting cone snails make snail-like insulins, and the worm hunters make worm-like insulins. So it's one of those, it's a very rare example where you can really track the compound to the prey. And so we don't think that it's typically used in defensive purposes.
Okay. The other question I had was looking at the total, and maybe, I don't know if you've done this type of research, but looking at the total venom package that the snails shoot out. Do they modulate that, like levels of different toxins based on the prey they're going after? Or like, how small, like?
I think that's still controversial in the field. There was a study published that still needs to be, replicated by other groups, where it was suggested that depending on the defensive or predation triggers, the cone-snail can release different toxins. It's a nice idea, but it hasn't really been confirmed by others. We do think that the snail preys on different types of fish and using different types of cocktails of venom, but how that's regulated is not very well known. It could also be that it's always similar things that are released, but they then just act differently on that particular prey or that particular circumstance. So those behavioral studies are kind of in their early stages.
Okay, yeah, that makes sense. And then, so looking at the, I guess there's a broad scope of, you said, animals that are using insulins to do, hunting, like to assist in their hunting. Are there other maybe you can't talk about it because it's early in the works, but are there other specific animals that you're really interested in?
In terms of insulin, so there have been a couple of studies suggesting that insulin-like compounds have been found in some snakes.
Okay.
Unfortunately, the sequences have not been made available that easily. So we're hoping that more data will come out in the near future, but there would be other venomous animals that use insulin-like compounds for prey capture. It would be surprising if of all the many hundreds of thousands of venomous animals, it's only cone-snails. So the snake studies have been interesting, and there are some interesting compounds in the genome of some snakes, but without finding the actual actual compound at peptide level, it's really hard to predict what it would look like. Is it a single chain? Is it a double chain? How many disulfides? So you really ideally have to have the venom and put it on a mass spec, and then it's not a straightforward molecule to identify by mass spectrometry. Other peptides are much more easily found. Then there are also insulin-like compounds made by viruses. And again, those have not been confirmed at the peptide level. So you see the transcripts and the DNA sequences, but you don't see, people have not found the actual peptides yet. So it's hard to know what the real thing looks like in the end.
Will you talk a little bit about the process of identifying the peptides? I mean, you talked about using mass spectrometry, but maybe start from like you go into the ocean and you get a snail.
So we go into the ocean, we get a snail, mostly by scuba diving, some by snorkeling. A lot of it is through local fishermen that know exactly where to find the snails. So we are actually quite terrible at finding the snails ourselves. So we go out for hours scuba diving and find maybe 10, and they come back with, you know, as many as you want. And then you go and you dissect the venom gland out of the animal. And then in the past, people would need a lot of different snails and a lot of venom glands to have enough material. And now the technologies have become so sensitive that from a single gland, you can sequence all the transcripts that are expressed at that given time in that particular species. And then you can also use the venom and extract the peptides and put that on a mass spectrometer. That process for insulin is not straightforward. So you first, especially if you want to know what the intact molecule looks like with disulfide bonds, and especially if you have multiple transcripts in your species. So we typically, you have to first do a run where you reduce all disulfide bonds, you chop it up with trypsin, and then you look at all the fragments of the different insulins that are in venom. And then in a second, and then you predict what the intact molecule would look like in terms of what would be the mass of the intact molecule with the disulfide bonds and the cleavage sites, because they can also be present or absent. The C peptide that connects the A and B chain could be there, it may not be there. And then you look for that intact molecule in venom. And then if you're really strict and rigorous about your research, just seeing that intact mass is not good enough. So then you have to isolate that intact mass and do the same thing again. You reduce the disulfides, you chop it up again, and you confirm that that intact mass of your insulin molecule is your actual sequence that you predicted it to be. And then you can say, this is the insulin, the mature insulin that comes from that particular transcript or gene. And for other peptides, it's much more easily done for insulin because of the two chains connecting and the multiple different cleavage sites and the different ways the disulfides could connect. It's quite complex.
So why did you, I mean, I guess I should ask you first, are you studying other toxins then within venoms?
Yeah.
Okay. Go ahead.
So we typically look at all the toxins. Nowadays, you go and find the species, like in this case, Conus geographus, or in the more recent paper, Conus kinotretai. And then you take the venom gland and you sequence everything. The transcripts, the methodologies are so sensitive that you really almost get every single transcript. And there could be up to 500 or 600 different toxin transcripts in each species. And then when you put that on the mass spectrometer, only the most highly, let's say the 80 or 90 most highly expressed toxins you would actually find by mass spec. And then those could be modified. So a lot of these toxins have different post-translational modifications. So they don't come as a single isoform, they come in multiple proteoforms, so multiple versions of the same transcript can lead to many peptides. And then we sit there and discuss which are the toxins we want to make and which ones do we want to test, because then the actual hard work starts. Looking at sequences is easy. Deciding what to take and what to pursue and spend time on, that's most of the time the hardest decision.
Yeah. Danny, how do you sort of, in your group, how do you think about that? I mean, are you working with other toxins? Actually, I didn't even ask you that.
Actually, we did, but it's for an effort that's quite different from this. So this is also another pretty serendipitous thing. So I happened to sit in a graduate student's thesis committee that is basically Helena's co-worker. His name is Michael McIntosh. So he's a medical doctor, researcher. They study quite a bit of different toxins that can target some neurological receptors in the way that can create non-opioid related pain relievers. And then we also work on some of those special toxins and then try to do some peptide chemistry on top of it in a way that try to further improve the property. So, but that's an effort quite different from our insulin efforts.
Yeah.
So I will say I just feel fascinated by all the things they can do. That's pretty far away from my my own expertise, but whenever I found that they can discover very interesting peptide sequences and then quite a bit of them with very special properties, and then it's pretty obvious for the insulin thing, but for some of those other targets, they all have a kind of relatively common kind of disulfide rich skeleton or structure, but they just happen to have mutations here and there, and then they can target very special receptors or some of the membrane proteins in the way that to have the effect. I just always feel fascinating about it. And then I also, because anyone can probably search for some of those lectures given by Toto Oliveira in YouTube in a way that, there are lots of things they have discovered so far. And in the old days, basically, they will run through HPLC, getting different fractions, and then try to do some assays to see what they can do. But nowadays, with what Helena just said, using sequencing and then, coupled with mass spec to confirm some of those sequences, the opportunity is way bigger. Now you don't need to just discover some of those most abundant ones. You can start to look for some of the interesting molecules, and it may be not enough for detector to see it from a HPLC perspective, and then we can now discover something pretty cool and then trying to do some chemistry in the biology on top of it.
Once again, you've segued into a question. Helena, I was going to ask
Lots to learn from Danny. Learn how he does that.
I swear we didn't meet before and do a plan of hey, I'm going to ask this, then you go here. But when you were speaking about the mass spec, and Danny just hit on it, that you're going to have these different abundances of these toxins, and it does seem like you might have a unique opportunity to find extraordinarily potent molecules, because you would expect the ones that are in the lowest abundance I guess it would depend on what their activity is, right? Because some maybe they just are synergistic with other molecules, but so I guess that adds another layer of complexity to when you are looking at sequences of how you decide what should we go for.
Yeah, and that's really hard, because there are so many things you could potentially study, and many of these toxins are ion channel blockers or ion channel activators. We recently started to look at GPCR modulators, because people have not done that thoroughly for toxins. So we felt like there's a niche we can explore. But it's really difficult to decide which toxin to pick. The advantage that is now emerging, because we have so many sequences and we have found a lot by mass spectrometry already, that we are now at the stage where we can predict post-translational processing in many cases. So we can go back to some of the transcripts we're interested in and say, based on what we found in Venom, can we predict what that toxin, what that transcript would actually translate into in the mature peptide. And for the latest insulin, actually, the cone-snail insulin, we never found in Venom, because we couldn't get enough venom of that species. And so we predicted it based on what we had learned on the Conus geographus insulin and made different versions of it. Yeah, but deciding which toxin to work on next is always critical. And through the insulin work, like our group has mostly decided to work on hormone-like toxins, because there the advantage is, let's say, like Danny said, you find many toxins, they just are sequences with disulfide bonds. You have no idea what they do. And so you could spend a lot of time trying to find out what those toxins do. Whereas if you see some sequence similarity to a known human hormone, then you have a good indication of what the target of that toxin would be. So you can immediately test it on the same receptor that the human hormone targets. And then the chances are very high that that toxin does something unusual that the human hormone does not, because the constraints it has during evolution are very different to the constraints we have when our bodies evolve hormones. So the venomous animal is really evolving it for predation, which is an injectable or absorbable version of the hormone we make in our bodies. So we are interested in these hormone-like toxins for practical reasons, but also for biomedical reasons now.
That makes sense. Are there people who do like panels of receptors for toxins and create databases? Like, is that type of thing happening?
Yeah, so what used to happen a lot in the field and still happens is, like, let's say you have your favorite receptor, like the sodium channel that is implicated in pain or in another disease, and you really want to find a good blocker or activator of that particular channel. You can screen venoms for that against that particular receptor or ion channel, and that is very commonly done. The same has emerged for GPCRs, so people just have panels of GPCRs and they throw venoms on. It's again not like that straightforward, because you normally take a mixture of venom components, and then once you get a hit, you have to go back and fractionate that mixture by HPLC or other methodologies, and then try to sequence that one component that gives you that effect. Sometimes it's not just one component, it's multiple components, so it also gets a bit more complicated. But people do that. If you have your favorite target receptor, you go and take some venom, throw it on, and see if you get a hit. It's more, you're likely to be more successful if you know more about the biology of that venomous animal or of venomous animals in general, because then you can go, okay, I want to find this ion channel blocker that is expressed at the neuromuscular junction. I should pick a venomous animal that when it ejects venom, the prey becomes quickly paralyzed. Because then you don't have to screen 500 different venoms. You focus on those 2 venoms that are likely to give you that toxin.
Danny, when you've been working with Helena on these projects, what's been the most sort of exciting discovery for you?
I will say the most recent paper we had that basically is related to the extended A chain there, where basically the additional residues we learn from the venom can basically totally compensate for, in the very beginning, what I mentioned, those lack of the C-terminal of B chain there. That is totally, that's a pretty surprising thing, because I would say that's something people would not have expected for insulin dipeptide. So in our body, there's also IGF-1 and IGF-2. Both of them do have longer A chain, just like these venom insulins, but these longer A chains, what people normally call it D domain, they don't have much role, at least as far as right now we know about what they are doing. They don't have much role about receptor engagement. So the fact that these, basically only 3 more amino acids at the end of A chain now, they can basically occupy a position where some of the residues in the deleted B chain would have done. That's totally amazing. And then something that's even more amazing, I would say, is back in 2019. So we had a paper in eLife where that was the very first time Helena reported even more sequences in addition to the original PNS paper, where the cone-snail sequence were revealed. At that time, it's more of a sequence, and then synthesis, and then some basic characterization data there. So there's no kind of deeper structural biology studies. We do have a modeling collaborator,
Brian Smith.
That he basically predicted exactly the same where we end up seeing it in structure. And at that time, we thought that was a little bit harder to understand, but basically that was exactly the case. So things like this don't happen that often, but this is kind of one of the exciting times.
I would like to add to that. I think that was really a beautiful interaction also between different fields. So we started sequencing more venom insulins, and based on what we had found, we knew that the B chain extension was missing, so likely the compounds are monomeric. And so then we looked at the sequences and just looked at them and said, what is the unusual feature about some of the other sequences? And just in an alignment, you would see, okay, the A chains are extended. There must be a purpose of that. It's not random, evolution is not random. So there must have been some selection pressure on an extended A chain. And these cone-snail species happen to be not related very closely to conus geographers. So there's also, it makes sense that distantly related species has evolved a new mechanism that is different to the first mechanism we found. And then we sat down together and said, okay, let's look at that extended A chain. And then Danny put it on a minimized human insulin and basically found that it could activate the receptor, which was quite exciting.
That's incredible. What, for you, just being in the field of venoms, what has been the most sort of, for you, the most exciting discovery that you've made?
I think the insulin was probably the most exciting. And at that very moment, like Danny said, I was still a postdoc in Toro Oliveira's lab, and people had studied conus geographers for a long time. It's one of those most studied animals. It's also the one that is known to kill people. There are not many fatalities, but if you do encounter that one in the wild, don't touch it. So people have been interested in this particular cone-snail for a long time. And then because I was trained during my PhD studies in mass spectrometry, I then found an insulin in venom. So I presented it at lab meeting. I said, there's something that looks like insulin. And the moment Toto heard it, he's like, drop everything else you're doing. And I was actually working on 2 other really exciting projects that I was totally into. And he said, you have to drop everything you're doing, focus on that now. And I think that has remained probably the most exciting discovery. And then more recently, we found another weaponized hormone that I'm particularly excited about. It's somatostatin. So we find these somatostatin-like toxins. It's been harder to communicate my excitement to others because people just know insulin. You just talk to your neighbor and you say, I work on an insulin toxin. You talk to the same neighbor and say, I found a somatostatin toxin. They go, It's not the same level of excitement you can create in others. But personally, that's also very exciting recent finding.
Yeah. What do you think, as you're, looking at the field, where do you think that the future is going with venom molecules?
Yeah, I think what is now emerging and people are appreciating it more. I think in the beginning it was just that venoms are a great natural resource. We can throw them on our favorite receptors and ion channels, and maybe we get a hit, and then we just focus on that one toxin. And now people have started to appreciate that by understanding the biology of the venomous animal, we can learn a lot more. And we have to assume that the compound we're looking at does something that we may not understand fully, but that we should further investigate. In the case of the insulin, it was fairly easy for Danny to immediately spot that this is an interesting compound because it has that and those amino acids missing. But then there are a lot of other compounds where we look at them and we don't really appreciate the beauty of what they do. So we really have to thoroughly study what they do. And it has become a lot easier with the great advances in structural biology. So we can really go in and look at interactions. Now we can predict interactions using AlphaFold. So it's really I think the field is moving forward at a very fast pace into the right direction. So it's an exciting time to do this work.
It really speaks to I think the importance of basic research, right? I think we all talk about translational research all the time. You know, we're making medicines, we're creating technology, but there's something to be said for just understanding what something does. So I think this is like a perfect example of that. Yeah. Danny, I was going to ask you a similar question. You know, as you're working with Helena and seeing these natural molecules that can be, now I'm going to go back to translation, but they can be translated into medicines. What do you think the biggest breakthroughs are going to be, or what are you most hopeful for?
So for sure, I think these monomeric insulins do have some special features that we can address some of the challenges in the glucose management for people with diabetes. So one of the kind of most exciting development in the therapeutic end of glucose management is the use of the closed loop system, where basically there's an algorithm to control what's the amount of insulin the pump wanna inject it into the body, and then use the continuous glucose monitor to be a readout to make the decision. One of the challenges still is basically the speed of insulin action. So that makes it quite difficult for the algorithm to make the perfect decision about what's the amount of insulin to inject in and then how much they should do it. So if someone can have a fast enough insulin and potentially with a relatively short duration that there's no overlapping dosing effects there, this could make the algorithm to basically further maximize the impact of such system for people with diabetes. And I think monomeric insulin can be one of the best solutions to tackle this question. So I think therapeutically, there's a huge potential for a molecule like this to make a huge impact. In addition to this, like what I mentioned earlier, I think some of the knowledge we learned from the features here can really be used to make additional pro molecules to study the signaling and then basically further understanding what we can do about the insulin signaling. This may still be pretty far away from potential translation, but one might think that insulin can do the metabolic action and the mitogenic action. And most of the time, when we are talking about glucose management, it's more about the metabolic option. So if one day we can really be like the GPCR field to create bias signaling, we may be able to generate a biased insulin receptor agonist in a way that can give us more of the features we like and not as much of the features we do not like. And this could be good in the long run as well. But in this case, we are still pretty far away from that.
Helena, is there anything that I didn't touch on that you think is really important to know about the field of venom research?
Not, I think in terms of the, because we're looking at a small aspect of it, which is mostly the insulin or hormone-like toxins. So I think we covered that really well. So I really think what I would want to get across is that it's always worthwhile to look at nature, even in a system where you think you understand almost everything already. I mean, it's one of the best studied systems, right? And you can still learn new things by looking at what nature has already evolved. So I think that's a good message to carry forward.
Thank you. It was a pleasure to meet you. And Danny, it was great to see you. Thank you both so much for speaking with me.
Yeah, and I think I will see you in person again, maybe in Vancouver, right?
Yes, absolutely. Whistler, yes. Are you going to be able to go, Helena?
No, probably not, but I'm hoping to see you in person or meet you in person at another meeting in the future.
Thank you for tuning in to this episode of Exploration Science. If you enjoyed this content, please like and share with your communities. As always, we appreciate your feedback as well as your suggestions for topics that you would like to see covered. Thanks again for tuning in.